So my name is David Fenyo, and I'm in the Biochemistry and Molecular Pharmacology department, and also member of the Center for Health Informatics and Bioinformatics. And I'm going to talk about summarizing several different collaborations that I've been involved in. The goal is to develop new both experimental technologies and also informatics technologies to better able to study protein-protein interactions. And this is collaborations both with the two proteomics labs here at NYU, which are headed by Beatrix Uberheide and Tom Newbert, and also with Rockefeller University with Brian Chait, who is running a mass spectrometry lab there, and with Mike Rout, who is a biochemist. And then it's also collaborating with Eli Rothenberg here at NYU. And so the idea is that with affinity capture combined with mass spectrometry, we can look at what different proteins interact with a certain bait protein. But there are always issues to determine what is a specific interaction and what is a non-specific interaction. And I'll talk a little bit about how we're trying to solve these issues to distinguish those. And then the idea is that once we have a set of proteins that we believe are interacting, then we can go and use super resolution microscopy to look at the details of these interactions. So first, I'll give a little bit introduction to mass spectrometry and how we're analyzing proteins with mass spectrometry. So we take samples, and then there are many steps to go from the actual samples to making peptides. And what we preferably want to analyze in mass spectrometry is peptides, because they behave well. You can separate them, you can ionize them, fragment them well to both identify and quantify them. And the two things, so what we measure in mass spectrometry is mass to charge, and at each mass to charge we measure an intensity. So the mass to charge we can usually with modern instruments measure very accurately. We can measure very accurately, and we use it to identify the peptides, and then we can roll that up into proteins and identify proteins. The y-axis we can use to quantify, but the quantification is slightly more problematic. The identification we can do really well, but quantification not so well. For several reasons. One being that if you have two peptides with different amino acid sequences, you mix them together and at equal amount, there's no way anyone can tell what their relative intensity will be nowadays yet. So there's no theoretical way to calculate that this peptide is, and also if you change the solvent conditions, you can even switch so that one is one order of magnitude higher than the other, and you can so that's the problem. So all the quantitation in mass spectrometry is to use different internal standards to be able to at least do relative quantitation. So the way we do identification is we take the sample, we treat it in, we usually extract the proteins from the cell, maybe separate the proteins, digest the proteins, usually with trypsin, and then separate the peptides and analyze them by each peptide by mass spectrometry. So we start with usually complex samples of hundreds, thousands, or maybe 10,000s of proteins, and then we digest these proteins to peptides to make the sample even more complex. So what we do is then we need to separate these peptides out so that the mass spectrometer only sees a few of them at one moment. So usually each of these experiments are at least 1 hour, but sometimes up to 4 hours long. So what we do is we collect all this information, and then we have a protein database where we have all the sequences that we believe are in the sample. And then we do sort of mimic what's done in the experiment and take each protein sequence from the database, digest it using the same rules as the enzyme, calculating all the fragment ions, and do these comparisons. And we have to compare each possible peptide to each possible fragment spectrum, and then we calculate scores and finally test the significance so we can set a cutoff at some threshold where we say that these proteins we can identify in the sample, and then the rest are have a high chance of being just random identifications. So one important thing here is that we really need to have the sequence in the database of our peptide that we're trying to identify. If we don't have the sequence there, we will miss it. There are some very that involves a lot of manual labor to analyze the spectra by hand and trying to figure out what the sequence is. But the problem is that there's often a lot of missing information in the spectra. So this is, and it also takes, could take a day or even a week for one spectrum to get it right. And so that we try to avoid. So what we want to do is we want to make a protein sequence database contains all the sequences, but of course we don't want it to contain a lot of irrelevant sequences, because then we just have a large database with a lot of noise, and we won't be able to get very sensitive identification. So usually most often what we do is we just take the reference, one reference protein sequence database and search that. But now it's been, with next generation sequencing being developed, we can actually use this on the same sample, do proteomics and genomics and whole genome sequencing or whole exome sequencing and RNA-seq, and use this information to create a sample-specific database. And Kelly Ruggles in my lab has developed a pipeline to do this efficiently. And if we do this, what we, in addition to just the reference sequences, we also will have somatic and germline mutations, alternative splicing, gene fusions, and novel expression. So these are the things that then we can look for in different projects. And I'm going to go back to one project where we use this for sequencing antibodies. So then quantitation. So what I mentioned was that quantitation is, so people have worked out detailed methods for identification, and we've also worked on this quite a lot to determine different methods to calculate the statistical significance of the results and so on. So this is, there's still work going on here for improvement, but it's much more mature than the quantitation field. What we have in quantitation is that we measure the intensity of a peptide. But what we're interested in is the concentration of a protein. So now we have all these steps in the sample preparation where we lose material. And of course, these losses are different, are protein and peptide dependent. So in a sense, if we could measure all these losses, we could calculate from the protein concentration what the intensity of the peptide would be. But we don't. But what we can use this for is if we do relative measurements, we can see where we get more, in which experiments we get more variation and how we can get less. So the sum here in this term is over the different peptides that can be from, each peptide can sometimes be from different proteins. So that complicates things, and in general, that's something that in the proteomics field people don't really deal with very much. So one way to do then quantitation, relative quantitation, is to repeat the same experiment for different samples. And here we have, the key here is to try to vary the conditions as little as possible. So we do try to do, and the assumption is that all the losses along here is the same. And then we can calculate the relative intensity, the ratio of the relative intensities, and get from that the relative concentration. People do this, and I would say it's probably mostly recommended for when you have a few samples, but people have actually done this where they've analyzed 95 colon tumor samples over 3 months. But you can imagine how that's a bit risky, because you will, in 3 months, a lot of things can happen and a lot of things can change. So another way to go is to metabolically label the sample. And here, the advantage here is that we metabolically label, and the only time that the samples are separate from each other is when they are growing, and then we mix them. So the rest of the workflow will be, the samples will be together, and the assumption is also that the heavy and the light isotopes behave the same way, which they usually do if you use carbon-13 and nitrogen-15. The other question is, does the heavy isotope affect the growth of the cells? And in general, for cell culture, the assumption is that it doesn't, but it actually does, especially when people have tried to make heavy isotope labeled mice, and they are very sick. Because there is there is an effect. But that's again, usually one assumes that there is no effect of that. Okay, so then the main way that people look at protein interactions by mass spectrometry is by doing an affinity capture. So here is an example. We have a protein complex, and one of the proteins we tag. So presumably we know maybe this protein C is the one that we know is part of this complex, and we're interested in which other proteins it interacts with. So this is, of course, in a complex environment. And so we usually have magnetic beads with antibodies against the tag, and then we pull out the protein, and we try to pull it out so that as much of the non-specific proteins are washed away, and hopefully the whole complex is kept intact. So, but what always happens is that a few non-specific proteins stay around, and we lose a part of the complex. That happens all the time. And so these are what people are trying to sort of minimize the number of non-specific and keep the complex intact. And so what then the next step is to do digestion and mass spectrometry to identify what proteins are there. So so in the Mike Rout's lab at Rockefeller, they've been developing to optimize how one does this, the affinity capture. And the way they do it is that they grind the cells down to very small pieces. Preferably, it should be pieces the same size as the the protein complexes. But they're not there yet, but that's sort of the goal, the similar order of magnitude. And what they do is they use liquid nitrogen to freeze the cells and then use these balls. This is a grinder that rotates, and then the cells get crushed with these balls. And then the next step is that all the protein complex behave slightly differently. So you really need to explore a lot of different possible solution conditions on where to, and also this is work from Mike Rath's lab, where they develop the robotic system so that they can test a large number of different solvent conditions. So what happens is that if you do one pull down and try to do it in different conditions and then just look on a gel for what happens, so you see that sometimes there are very few bands, so only very few proteins stay. Then during other conditions you get a few more proteins. But then yet other conditions you get just a lot of background. So the idea here is that to try many conditions and then select out a few, both ones where you get very few proteins, they're really the tight binders, but then also some where you can see more proteins. So if we look at one example here, this is from the nuclear, pulling out proteins from the nuclear pore complex. We again see that sometimes we just get 2 proteins here. Here we have a third one, and then here in this case we have a few more. So that's and one really has to do this for every protein complex. So here is just example of a few protein complexes, and showing both buffer, salt, and detergents. And one can see that the ideal conditions for these different protein complexes is quite different. So definitely this has to be done in every case, ideally. So then in connection with this, so of course, if we're going to do this for every protein complex, then we need to manage this information and store it in a database, so that, because it could be interesting then to, if there is a lot information collected, to mine the data and see in general what kind of conditions are useful. So Sarah Keegan in my group has been developing a system where one through the web can submit gel images and information about that. And then the system will process the gels automatically, cut them up and then into lanes, and then recalibrate both the mass scale and also the intensity scale, so that we can, if there is an internal calibrant in the system. So another tool that one can add to this is chemical crosslinking. So chemical crosslink can be used to stabilize the protein complexes. And if we want to just stabilize the complex, then one can use very low concentrations. And here the idea is not to find exactly the site of interaction, but just to make the complex more stable, so that we can wash harder and remove the non-specific interactors. So again, what we do is we go through the same process that we digest with an enzyme, analyze the proteolytic peptides, and look at the MSMS spectra to just identify the proteins. So this is also actually collaboration with John LaCava, where he looked at the nuclear pore complex, both did a pullout, without cross-linking. So those are the black dots. And then with cross-linking showing with the red dots. This work was done by Betul Oksuz. So what we then did was, what one can see here is that without the cross-linking, we get a few proteins out. But when we do just a little bit of formaldehyde cross-linking, we almost get the whole nuclear pore complex in one affinity pull-down. So another, so far what I've talked about is when we use, we genomically tag a protein and then have an antibody against the tag. But that's sometimes difficult, and sometimes it's better to make antibodies. So another project together with Mike Rout and Brian Chait is about how to make antibodies that are very specific, and it's easy to do. So one thing in camels and llamas, they have our type of antibodies with heavy and light chain, but they also have a single heavy chain antibody. And the nice thing here is that if we can determine the sequence of this, the variable region of the single chain antibody, then we can make very large amounts of the variable, and we don't have to in bacteria, and we don't have to worry about combining light and heavy chains. So that's so the idea here was to combine sequencing of the bone marrow. So we take the llama that has been injected with, so in this case with GFP. We take the bone marrow and do PCR of the variable single chain antibody, variable region of that, and then do next generation sequencing. So we get a sequence library. And from the same llama, then we take the crude serum, and from there do an affinity pull down of the highly specific antibodies against the GFP in this case. And then we analyze that by mass spectrometry. And now we make, from this, from the DNA sequencing, we make a protein sequence database that we can then search with the mass spec data. And then finally, we combine together the information to select which antibodies to look at. So this is then one example of the viewer. Again, so Sarah has developed a web interface for this, so that we can see now the sequence of the translated sequence from the next generation sequencing, and then we see which peptides we observe, and then the parts in red are the CDR 1, 2, and 3 regions. And then we can select out exactly which antibodies we should clone and test. And in the case of GFP, what happened was that we had 44 that we thought were high confidence clones, and 26 out of those turned out to be GFP binders. And then this process is quite fast now. You can probably do this in the whole turnaround in a few months. And the nice thing is that you get several highly binding proteins. And the other idea here is that what we did was look at where on the GFP these antibodies bound. And most of them bound in the same region, but some bound on opposite sides. So then one can, of course, cross-link these and start with having two strongly binding antibodies, getting one extremely strongly binding antibody. So another project, also in collaboration with Rockefeller and with also the main PI is Mark Museling, is to look at the HIV interactome. So here we're then applying some of these methodologies. So what they wanted to look at was when the HIV infects cells, which proteins, looking at the envelope and the VIF viral proteins, then trying to figure out which proteins in the cell do they bind to. So here, the problem with HIV, of course, it's a very compact genome. So if you try to put a tag in anywhere, it usually loses viability. So what they worked out was that one can do a random insertion and then have these different clones compete against each other when infecting cells. So during the whole life cycle of HIV. So this the figure here just shows where they insert. So they insert randomly, but there are definitely hotspots, and not every base you can insert equally well. But we start out with, this is what we start out with. But then if we let these compete against each other, already after a few days, there are much fewer of the insertions that are kept. And then we see that less and less, and then by day 6, we pretty much only have 2 different sites. So these are 3 different replicates. And we always get the insertion in these different, these 2 sites. So then the idea was that from this we can then, this was just a 5 amino acid insertion, but we can then do insert 3x flag in there. So then here now, so I mentioned already before that there's a question of specific and non-specific interactors. So what happens, of course, when we do affinity mass spectrometry, when we have to disrupt the cells and pull the protein out. So definitely a lot of proteins that are not naturally in contact with our protein of interest or our complex of interest will come in contact with it. So we'll have a lot of spurious interactions that way. So one way that was suggested by Brian Chait to separate specific and non-specific interactor is to use heavy isotope labeling. So the idea is that we have our tagged HIV strain and we grow it in light medium. And then we have our the wild type HIV and grow it in heavy. And then we mix those. And then what should happen when we do the immunoisolation in the next step, the ones that are the specific interactors should only come from the tagged one. And so we should only see for those peptides the light peptide. But for the ones that are non-specific, we should see an equal contribution of specific and non-specific. So, and this is roughly what happens, but it gets, of course, a little bit more complicated when you do the experiment. We do have, so in this case, this is envelope and VIF. So if we look at the IDERT ratio here first, we see that there is a lot of proteins around a ratio 0.5, but of course not exactly 0.5. And then there are proteins at close to 1. But what we also have is proteins in between here. And then for WIF, the situation is slightly more complicated. We don't have very many really strong, close to 1, but we have, again, this region in the middle, and then we have the ones around 0.5. But in this case, we already saw that here it's a little bit of a shoulder here, but which is bigger for the for WIF. So and actually, so this the blue histograms here are just assuming that we have equal equal intensities. So we have the non-specific situation, but then we have the CV is larger than 0, so that we have some experimental variation there, and then we calculate the ratios. So that's just a very simple model trying to analyze that. So if you looked at that more in detail, so 2 things for the non-specific interactors. So ideally we would have them all at, so now in this case we have both reverse and so what we do is we either grow the wild type in heavy or the wild type in light, and the tag one. So we always make 2 measurements. So in this case we have, if we have a low CV, they will be around a small spread around 0.5, and of course if we increase the CV, there will be more of a spread around 0.5. The other thing is that we try to load the same amount of material all the time, but of course that's not always possible. So there usually is a shift. So if we have this imbalance, we see that this, instead of being at 0.5, this will move towards 1. Which makes it more difficult to separate things, but so one should definitely try, but one can definitely compensate for that also. So for the specific interactor, we have the same issue with the noise that we have. If we have low noise, they will be very close to 1, otherwise we get a spread. But the other thing we have here is exchange. So we can have, if during the time of the experiment we have proteins exchange very fast, we can then actually calculate what the IDERT ratio will be as depending on what the reaction rate is, what the exchange rate is. So, and we can also, depending on how if we assume that there are a lot of non-specific, a lot of specific but fast interactors, we can then get different distributions. So this is just assuming the same, starting with this case, but assuming that there is exchange. So we can, if there is a lot of exchange, we can also get a distribution around 0.5. That is now, I don't know if we can see that, but it is asymmetrical. So that would, that could explain this shoulder. And we could also explain why we have this background in between. So these are probably fast exchanges. So yes, so that was the end of the first part. How am I doing? Oh, sorry, I'm running. Oh, no, am I? You're fine. Yeah, I'm fine. Okay, good. Yeah. So the second part I'm going to talk about is now how we're using super resolution microscopy in collaboration with Eli Rothenberg to look at more in detail the interactions between proteins. So as you all know, there's a limitation in light microscopy. So if we want to look at a small feature like a protein, then, but we're limited by the wavelength of the light. So in this case, if we have 300 nanometers, we'll see a 300 nanometer blob. But what people have pointed out a few years ago is that if you have an isolated particle that emits the light, and you have long enough time to collect a lot of light, then what you can do is, even though the width here of this is 250 nanometers, we can determine the centroid much more accurately. And in this case, they actually, by collecting a lot of photons, could determine it down to 1.3 nanometers. So this is sort of the background to how one can go beyond the wavelength of the light with the super resolution microscopy. So, but now in a biological sample, it's crowded. So we don't have the luxury of observing each particle separately. So the trick is to excite the protein so that only very few of them will give off light at the same time. So in this crowded environment, what we do is we make these fluorescent molecules blink, but a very low probability. So at one time we just see one, then the next time we see another one, and so on. And then we can we collect the whole video, and then we can put together the image so that localize each of them. And in this case, we can't spend that much time, so we can do it probably down to 20 nanometers. So this is a project where we're working with Eli Rothenberg, but also with Mario Delmar and Esperanza. So they are very interested in the intercalated disk and seeing how the proteins there interact with each other. And especially they were interested in two proteins. One, plakophilin 2, that was known to be a part of the desmosome, and connexin 43 that was known to be part of the gap junction. But there had been some indication that these two proteins actually interacted with each other also, even though they were supposed to be classically in two separate parts of the intercalated disk. So if you look at first an image of this, the intercalated disk in regular microscopy, we see it's quite blurry, but then we can go in and do the super resolution, and then we can see much more detail. And so this is the intercalated disk here. So this is one cell, cardiac cell in this direction here, and then another cardiac cell in this direction. So they interact along here. And then we have the plakophilin 2 and the connexin. So then just zooming in on one part, regular microscopy and super resolution microscopy. So the difference here, what we see, so that with the regular microscopy, we might observe that a group of people are co-localized. But I mean, the question is, what does that really mean? In this case, there's very little interaction. But then with super resolution microscopy, we can see whether there is some meaningful interaction between people. So what we did was to then extract the information about these clusters. So things like the area, circularity, all sorts of things, anything that you can think of that one can observe. And what we saw was that there seemed to be two different regions. One is either there seemed to be some background where, independent of the area, there was just a little bit of co-localization. But then there was another group where there was usually a lot of overlap. And the overlap was, so it was between 50 and 100% overlap in between connexin 43 and plakophilin 2. So then the obvious question here, is this just random? Or is this something? So what we, then they did also another experiment where they silenced ankyrin G, which is another protein that was known to be involved in this. And what happened was that the areas increased. There was a loss of circularity, and there didn't seem to be this separation between either either not overlapping or overlapping a lot. So what Sarah Keegan then did was to do Monte-Carlo simulations on this. So now these are the synthetic images that we create in the computer that are supposed to be, so we take the distribution of areas, we take from the experiment the distribution of areas, the ratios of the axis of the circularity, and then sample from these distributions and do simulations. So both for connexin 43 and plakophilin. So the idea here is that we take some of the properties of the system, but we don't put anything in about overlap. So they're randomly placed in the giving rise to the same density and having the same area distribution and so on. And then we look at what happens to what fraction of particles will be in these two regions. So what we see here is that for the ankyrin, for this area being between 50 and 100% overlap, we see that for the untreated, we have the experiment up here, and the simulation from the completely random case, there's no interaction, no attraction between the two proteins. Then we definitely cannot explain the experiment with that. And the same with the ankyrin G silencing. But if we then introduce an attraction, so that we first lay out the plakophilin 2 and then increase the probability that connexin 43 lands on a plakophilin compared to landing anywhere. Then we get we can explain the experiment that way. So we can, and the other thing that we see is that so this here, sorry about the size, but this is the strength of the interaction, so that the probability that they actually land on top of each other. So what we see here is that we actually need a much stronger attraction in the untreated case than in the ankyrin G silencing case. So now what we can do is instead we can, as we suspected from looking at the data, the ankyrin G silencing introduces somewhat of a randomness, but not complete randomness. So that's the conclusion. So as I said, so this was a summary of several collaborations, both within NYU, with Beatrix Uberheide and Tom Newbert on the mass spectrometry side, and with Eli Rothenberg on the microscopy side, and also with Brian Chait and Mike Rout's groups at Rockefeller University. Thank you very much. Any questions? No.
So for the nanobodies, you mentioned that you checked those and you found, I think, 20 that had a strong affinity for the GFP from those llamabodies. Did you also check specificity? I mean, if you tried them like in a Western or immunofluorescence, you also get not only affinity but also the selectivity?
Yes. No, so we do get very good selectivity on several of those. I don't remember, not all of those probably, but a lot of them. Yeah. So they have I mean, they go through a whole range of different tests that they're doing. Yeah.
So one of the difficulties in doing purification and identifying co-purified proteins by mass spec is that if the tag protein is associated with multiple complexes, there's really no way of knowing which components are together and which ones are in separate complexes.
So what, I mean, I guess one thing that people do there is to do the reciprocal pull down. So you find some and then you tag those and but probably to really figure that out, you have to do microscopy.
Is there any biochemical way of, separating these different, purified complexes?
I mean, there are. People have tried like gel filtration or different things like that, but I don't think very successfully.
Right. I mean, they tend to be very large, and there's probably going to be a lot of overlap.
Yeah.
But yeah, that seems to be a major consideration. IP complex. It's like not all.
I mean, I would say that you get some information from mass spec, but you have to go further and then do other experiments also.
So in the intensity variations from peptide to peptide, you mentioned that even a heavy isotope can actually influence the ionization probability of some of the features, so that they're not a perfect match, but you assume that they are.
So in the beginning, the first reagent that people used deuteriums in a hydrophobic group that interacted with the column. So then you would have the heavy and the light slightly separated, which was very inconvenient. But then when people used carbon 13 and nitrogen 15, that is much better. That doesn't happen anymore.
My question, though, is about whether, for example, suppose you have a peptide and then you have another peptide that's identical, just have one additional amino acid. And let's just say part of it's not a charged amino acid. Are they likely to be pretty darn similar, or is there absolutely no correlation? You have just one amino acid differences.
For small peptides, I would say it's very, I mean, if it's alanine, it's probably fine. But if it's any thing else, I think it's, I mean, even if it's a hydrophobic group, it's not good. It's difficult to say. When you have a whole protein and maybe one amino acid changes, and that might be okay.
Because you have all the...
Yeah, because it's the best thing is to just compare the same peptide in different samples or the same peptide to an internal standard. The other thing that is very difficult because of this is stoichiometry for protein complexes. That's extremely difficult to determine.
So one more question. Not directly related to this, but I'm wondering if there are maybe a lot of small peptides that exist in the cell, like microRNAs or something that we haven't really detected. Is there a way to use mass spec to detect small peptides that may be potentially functional?
Yeah, so you mean like naturally? Yeah. So people have done that, especially in the brain, looking for different peptides that are neuroactive. So there's quite a lot of work in that field, not by a lot of groups, but by a few groups. So that the other thing is small open reading frames in general. And there has been a little bit of work. Actually, I'll have a visitor who just in 2 weeks who just published something. So if you're interested, I can organize you meet him.
Yeah, I mean, that just, maybe because half-life might be very short, but I know like half the genes have open reading frames, every single open reading frame. So the question is...
Yeah, so he's using, I think, a combination of mass spectrometry and ribosome sequencing to find these small open reading frames. Thank you.